because he wants to have more time for talking. And just I would say he doesn't really need my introduction. You can Google and find out about Jeff. He's a graduate of MIT, and he has always, in fact, the entire career life, I think he has been in Boston, and he has received several awards, several novel papers actually in Nature. As I said, you can Google and find about his background. Again, I'm quite. I feel it's a quite honor. I thank you for accepting our invitation, coming here. He has been giving 2 talks yesterday at Faculty of Medicine, and today had a session with our students in the afternoon, and a few visits, and now we are looking forward to your talk. Please welcome, join me to welcome Dr. Fredberg.
It's a pleasure and an honor to be here. Thanks for the invitation. Let me just start. I just want to make sure I kind of understand the audience. How many undergraduate students? Graduate students? Postdoctoral fellows? Okay. I think I get the idea. So, so this is kind of a bioengineering thing. So it's going to be a lot of engineering, a lot of bio. So let me ask you, what organ system expresses the largest surface area? Anyone have a guess? The faculty would be fine. You probably students. So, there's lots of things in the body. What has the biggest surface area?
Skin.
Skin. Okay, skin is a good guess. It's 2 square meters. It's about like a tablecloth. Okay, who has another guess? Lung. Okay, lung. Okay, getting them in the right order. Pulmonary epithelium, it's about a tennis court, about 200 square meters, 140. So imagine a tennis court, and the lung has about 200 ml of blood in it. So that's about a martini or a wine glass. So imagine taking 200 ml of blood and spreading it over a tennis court. That gives you an idea of the dimensions in the lung. But it's not this, and who has another guess?
Kidney.
Kidney. Well, I don't have kidney on here.
Intestines. Intestines.
I couldn't.
Intestines.
Intestines. Actually, I don't have that number here. But actually, the next one, endothelium, like all the blood vessels everywhere. If you add up all the endothelial surface area, that's 1,200 square. That's 6 tennis courts. That's a lot of area. Okay, and this is what, and these happen to be endothelial cells. So I'll show you better pictures later. So they line all the surface of all the blood vessels, right? And then an epithelium line everything that kind of is like in the lung surface. So these are monolayers, and they're big. They're big, and they're big for a reason. It has to do with transport. And it turns out they're plugged into biology everywhere. So, for example, morphogenesis, like when you have an embryo and it has to divide and spread, these are monolayers, layers of single cells. Growth, migration of cells, barrier function, like in the lung, the epithelium has to be a barrier between the outside, the air and anything that's in the air, and the body. The same thing with the endothelium, has to be a barrier between the blood and the tissue. Lung injury, for those of you who are interested in lung, I'm a lung guy. So there's respiratory distress syndrome, where there's a problem with barrier function, and ventilator-induced lung injury is a big problem. There are people on ventilators. The ventilators can do more damage than what the disease does. It can cause disease. Leak, where you get inflammation, swelling, pulmonary edema, inflammation, repair, vascular disease, airway disease. A lot of you don't know about epithelium mesenchymal transition related to something in growth and cancer. Carcinomas, it turns out carcinomas are 90% of all cancers derived from epithelial cells. And then metastasis, invasion, these are bad things having to do with cancer, and it turns out cancer cells tend to move in chains, ducts, strands, and clusters, not individual cells. So all these have to do with how monolayers behave, monolayers like this one. So each of these, it turns out, what makes it, what I'm particularly interested in, these are not individual cellular processes, and there are plenty of people who study individual cellular processes. These are collective. It has to do with how the group behaves, not how the individual cell behaves. And the particular thing, I'm a mechanical engineer by training. I'm interested in forces and motions, that sort of thing. I don't know very much about chemistry. But the question is, when something like this moves or spreads, what are the physical forces that makes it move? Oh, by the way, feel free to interrupt. If I don't get to the end, it's okay. Okay. The problem is, I'm going to show you, there are physical forces involved when these cells crawl, but the thing is they're invisible. You can make images of structure, but how do you make images of forces? That's a lot harder to do. So here's a junction between two cells. There are forces there. The question is, what do they look like? So I'm going to tell you a story in 5 acts. I probably won't get to all 5 acts, but these are some of the players. Actually, this is an old picture. It's a lot of these people have gone from my lab. A lot of new ones came in, but a couple of key people I want to acknowledge. This is Jim Butler. He's a mathematical physicist, and nothing in my lab happens without his participation. He's really something very special. This is Ning Wang, who was one of my trainees. He's now a professor at Illinois, who's done very, very well in cell biology. Javi Tripats in Barcelona. Some of the things I'm going to show you are things that he did when he was in my lab. And this is Donna J. Tombe. I'm going to show you some work of him as well. Okay, so this is the title. It's not the title I gave Zara. I changed it. I think it was mine. So I called it Crowding, Tugging, and Jamming, Mob Control, and Collective Presence. So I called it that for a reason, you're going to see. So the first act is a tug of war, correcting the rugged landscape, and so on. And I probably won't get to these. So what's the tug of war? Okay, so this is a layer of epithelial cells. They're actually called MBCK cells in the kidney. And I'm going to play a movie. There they go. And this is called a wound healing assay. So there's no cells here as if there's a wound. And here are these cells. They're programmed to heal the wound. Okay, this is a traditional kind of wound healing assay. And some of you have probably seen movies like this before. So if the cells are moving, the presumption is that there have to be physical forces involved. These cells, they're crawling across the substrate. Maybe they're pulling on each other. And the question is, what are the forces? Oh, sorry. I'm going to move these up. Okay. So it turns out motions like these have been recorded for just about 99 years now. This is the first paper I know of, 1914. So this is not news. We've known this for a long time. And the presumption has been there have to be forces involved. And the question is, what's the relationship? If any people backgrounds in mechanics, so the relationship between motions and forces in any system is called mechanics. That's the field of mechanics. So what are these like? You could poke, is this a solid-like material? It looks like it's flowing. Maybe it's fluid-like. And the question is, what is it? Well, people have been wondering about these kinds of questions for a long time. So there have been 3 frameworks that are in the literature, if you read the scientific literature, and interestingly, they're contradictory. They all cannot be right. So here's one, and this is actually very current in the last 10 years or so, and it has to do with leader cells. The notion is that there's specialized cells at the front that are called leader cells that pull the other cells that are behind them. So the physical picture would be, you get in bed, you grab the edge of the blanket, and you pull it up under your chin. So the idea is that these leader cells are pulling and they're dragging the cells behind. So that's a pretty simple idea. If that's true, that means that these cells here are in tension. They have to be in tension, right? When you pull up the blanket, it's going to be in tension. And so that's been thought to be important in wound healing. And a lot of the literature, a lot of people are studying these specialized leader cells. Another idea that's in the literature, mostly from engineering, physics, people who model this. They say, no, it's not about the leader cells. It's actually each of these cells is self-propelled. So the model would be like cars in traffic. Each one propels itself. One isn't pulling the other. Or soldiers walking in a column, right? Each one is self-propelled. So if that would be the case, then the state of stress here would be there's no intracellular stress. They're all just moving along, pushing themselves. In the cancer field, people say, well, no, it's not this and it's not this. It's actually this proliferation of cells back here. Cells dividing, proliferating, and they're pushing the front forward. So that's, the colony expands and this front gets pushed forward. So it actually, these cells are in compression. They're pushing the things in front of them. So, let's see, these three are mutually exclusive. It can't be all three. And so there's no consensus, and even the sign, you say, well, what is the stress in here? It's guesswork. You don't know. And the reason is because the forces have been inaccessible to measurement. So what we needed to do is make these forces visible. And to do that, we needed some new experimental tools, and that's what some of the guys in my lab did. You know, by the way, you're only as good as your students and trainees. I did nothing. They're the ones who actually came up with these things. So here was, here's the same MBCK model there, and it was actually Javi Tricat. He's a guy that came to me from Barcelona, and he was interested in monolayers. I was not interested in monolayers at all. We were interested in smooth muscle because I was interested in asthma. And we had developed some ways to image forces in smooth muscle cells. He wanted to figure out how to do it in monolayers like this. It's very technical, it's very mathematical. I'll show you later how it works. So he worked with Jim Butler, and they figured out a way to actually image the forces that these cells are exerting on the substrate that they're crawling on. I'll tell you how it works later. So when we finally got this to work, we were shocked at what it looked like. Because we expected there would be some, whatever whether it's tension, compression, whatever, we thought it would be smooth. And so here's a plain envelope, plain vanilla group of cells. This is actually an image of what are called tractions. So these are the traction forces that these cells are exerting on the substrate. And it's color coded, so black is 0, so there are no forces here. The warm colors are cells that are pulling themselves toward the left. And actually the blue are cells that are pulling themselves to the right. And the first surprise is that it's very heterogeneous. We thought all the cells, they're all moving more or less in the same direction. We thought there would be some smooth distribution of forces, and it's not. It's very heterogeneous with a lot of cells pulling the wrong way. It's a mess. This is kind of the mob control I've been starting to work into. And if you actually play the movie, this is the movie, you can see they fluctuate. And so even you can't say that any one cell is pulling in one direction, because a little while later it's pulling in a different direction. So physicists call this dynamic heterogeneity. The forces don't seem to be related to any particular structure, any particular position, any particular cell. It's fluctuating all over the place. Okay? So that's dynamic heterogeneity. So that came as a surprise. You can see there's mostly red at the front edge. So these cells at the front, they are pulling. So they are specialized. They're leader cells. But it's not the whole story. And it turns out we're able to show, I'm not going to go into it, but we show in this paper. The leader cells basically are unimportant in the overall force distribution. It turns out what's happening behind. There are thousands and thousands of cells back here, and they contribute way more than leader cells. Okay. So here's another picture to kind of emphasize how heterogeneous this treatment and how the fluctuations are so important. Here's a piece of, this is bare gel, and you're going to see a monolayer of MBCK cells sweep from the left to the right, and over here is going to be a graph of the stresses, the tractions at that point as the cells sweep by it. This is my favorite graph ever. And so that's the background noise, and again I'll show you how this works later. here come the cells. I mean, it's just all of these it's all about the fluctuations. We had thought that there was going to be some kind of smooth variation with maybe some small fluctuations on top, but it's not. It's huge fluctuations on a small baseline. Turns out there's a positive baseline here that shows that there's tension, but the big picture is it's about the fluctuations. So this came as news. Turns out to be very important, and it leads to the last part of the talk that I'm not going to be able to get to. But maybe I can tell you at the end what that.
But wouldn't you expect that if the cells are moving at different speeds and they're sort of married together?
Yes, you might. We didn't anticipate, but it turns out it goes. You're right, but it goes way deeper than that. I'll tell you the conclusion at the beginning because it won't be clear at the end. It turns out that the cells are in a state that's called jamming. This is something that's very topical in physics. You ever go to the supermarket and there's coffee beans, and you open it and they don't come out, or they get jammed? That's jamming. You bang it, out they come. We have very strong, and what's characteristic of systems that are jammed or near jammed is that the forces show this. Even inner systems that are jammed show this kind of behavior. So it turns out cell jamming is now becoming a big thing. We wrote the first paper on it a few years ago, and this is a signature of jamming. There are other signatures having to do with the statistics of this, which we don't want to get into. Everyone's eyes would glaze over, but you're not wrong, but it's way deeper than that. It's more complicated than that. Or even, I'll give you an example. You've all had the experience. How many people have been to a hockey game? By the way, the Winnipeg Jets beat the Bruins the other day. My Boston Bruins. I was at the game in Boston on Saturday. So when the game gets up, everybody goes for the exit, right? And there's and it kind of converges, and you're jostling against your neighbor, and I really want to go that way, but you get swept this way. That's jamming. It's the same kind of phenomenon. Okay. So the conclusions are: it's all about the fluctuations. The action is far. Here's the leader cell, but there's plenty of action behind the leader cell. Okay. Oh yeah, no, I didn't go back. And look at this. This one is kind of, I alluded to this. This group of cells here is the same as this bunch. It's more than one cell. They're moving to the left, but their feet are pulling to the right. So it's like they're trying to go back where they were, but they're being swept along by the crowd. So what's that about? So it turns out in people who study cell migration, which are people in development and cancer and so on, how does one cell get to another place? These are the mechanisms that we know about. Chemotaxis means cells follow some kind of a chemical gradient. They follow their nose. Durotaxis means they can actually feel what they're crawling on, and they want to typically go to the stiff, soft here and the stiff there. They want to go to the stiffer region. And haptotaxis means they have differences of adhesion. They're more adhesive this way than that way, so they tend to go adhesion gradient. Well, this phenomenon is none of those. It's something else. Somehow the cells, even though they're pulling to the right, they go to the left. So there's some other phenomenon going on. So I haven't shown you the whole picture. This is only a part. This is the forces the cells exert on the substrate. What about the forces they exert on each other? Right? Again, when you're in the hockey game, you're elbowing the people next to you. But this is just the forces of your feet on the ground. So this is where Don and Jay Tombe came in. He was a postdoctoral fellow who came to me about 3 years ago. So the physical picture is something like this. Here are the cells, and these forces here correspond to these arrows of the stresses that the cells exert on the substrate. But then they're pulling on each other. So you get this kind of picture of a tug of war. And once you have this picture of a tug of war, it becomes really easy to understand, because anyone here, if I said this is one unit of force, another, and I asked, what's the distribution of tension in the rope? Well, it's simple. The forces have to add up. So it's 0 here, 1, 2, of how we, before I show you what those forces look like, the history of this, and how it works. This whole field was came from a guy named Cyril Harris in 1980, and what he did is he put cells on thin silicon sheets, and he showed that the cells actually could make the sheet wrinkle, and so this was a big deal. It showed cells generate force, almost, yeah.
The question, that tension measurement, is that in one direction or is it in both directions?
I'm going to show you, we get the complete the one I showed you is a vector. I only showed one direction, just for imaging purposes. You can get both directions, and the stresses in the cells are actually a tensor, and we can resolve the complete tensor. I know a lot of you don't know what tensors are, but I'll explain when we get there. But we get the complete state of stress everywhere. I'll show you that. So this was the first study, and it showed the existence of these forces, cell-generated forces, and it was qualitative, totally qualitative. You look at the wrinkles in there, and it must have been a force. It turns out a guy named Micah Dembo, a really smart guy at Boston University, he showed that using something more complicated than this, you can actually visualize the forces, I'm going to show you this, and quantify them. But it turns out the mathematics of doing this were horrendous. You need supercomputers and so on. It was then Jim Butler in my group who showed you can do the same thing as Dembo, but way, way, way simpler and way, way faster. And he made the code available to everyone. So now everyone uses Jim Butler's code. So it simplified the whole process. Instead of using a supercomputer, you can use a desktop computer. And it democratized the process because we gave the software away to everybody. And here you can get, actually, this is a physical map of the forces that a cell is exerting on the substrate. So the forces here, and it's pulling towards the center. And then Ramakrishnan in my lab showed how you can stretch cells and measure these forces. But then with this talk was actually the collective behavior. That was much harder. It was Tripath who came up with this, how to do this for monolayers and the idea of tug of war. That's the picture I showed you. And then Donna J. Tombe, this is what I'm going to start to tell you about, figured out how to get the forces between the cells. And there's several other things I'll go with. So this is how the technique actually works. We have a gel. Typically it's polyacrylamide, you can use PDMS, and it has fluorescent beads, these little green dots which represent fluorescent beads that we put just under the surface, so they're half micron. We use a different kind of fluorescent bead that down about 2 microns down. These are used for correction of stage drift. But what happens is when the cell, this is a monolayer cell, when the cell pulls on this gel, it deforms the gel, and we measure how much these fluorescent beads move, and so we can get a measure of the deformation of the gel. And then if you know how the gel behaves, and we know how the gel behaves, and you know a little mathematics, you can say, well, what were the distribution of tractions, these tractions, that caused that distribution of deformation? It's called an inverse problem. So you can solve that. So that's what Tripath and Jim Butler did. By measuring how these beads get deformed, when you pull on them, you can figure out this traction and its distribution. And it turns out it's almost as simple, and I told you already, if you know the traction forces, these red arrows, which you get from the beads, you can just add up the forces to get the intracellular stresses. So that's how it works. Okay? It's very straightforward.
So you just take a movie of the plate?
You just make a movie.
Make a movie and put the
You just make a movie, and you can actually, what you need the initial positions of the unstressed position of the cells. You can do that ahead, or then you can trypsinize, take the cells off, get that, and then this is easy to do.
Yeah.
This is easy. We do it every day. New people come in the lab. In a week or two, they learn how to do it. This is the math behind it.
Yeah.
It took some doing, but that's done. You look it up, and it's just the algorithm. So those are this answers about your question. We actually get this is a vector, TI, so it's in the plane. We get the vector, and from that, it turns out this is a differential equation. When you solve that, then you can get everything you want to know. And it turns out, this is really important, it turns out to be virtually independent of any assumption. You don't have to assume anything. This is not a model. This is not a theory. This is a measurement. And all you have to assume is that the gel is elastic, and you know the elastic property. It is, and we do. I say almost model-free. There is some subtleties. The subtleties are described in this paper that just appeared, for those of you who are interested. But for practical purposes, it's this. It's just this. Okay, I'm going to skip that. So, Act 2. I'm supposed to finish when? So,
6:20.
okay. All right. So trucking the rugged landscape. This was an important discovery. It's something we call lithotaxis. Okay. So this is now an endothelial monolayer, and we were interested in about, okay, we can measure the traction forces. What about the intracellular forces, the cells that the forces that one cell exerts on its neighbors? So actually, here's a map doing the process I told you about. This turns out to be a map of the intracellular stresses here. These are actually the tensor stresses. So if you can, it's kind of shown here in perspective. That corner is that corner, this corner is this corner, that corner. Everyone gets that? And it's really striking because when you look at the tension in this monolayer, it's very heterogeneous. It's not like the plains of the tundra. It's not like Vermont, even with rolling hills. It's like the Himalayan mountains. You get these huge peaks of tension, and then not very far away, a little bit of compression, and then another huge mountain range of tension. It's, again, tremendously heterogeneous.
But the landscape changes.
It's changing all
the mountain can move.
It moves, it disappears, a new mountain range appears, and but this is very typical of what we see. It's very typical. And this mountain range it's created by a pack of about 30 cells that are one's pulling on the next, pulling on the next to cause this buildup of tension. So we call it I don't know if I wrote it here. It's like a tension pileup. This tug of war, and you get this huge pileup of stresses. It's cells acting cooperatively. A bunch of cells all decide to pull in the same direction at the same time.
Is it also true to say that it's related to the orientation of the cells?
It is. It is. We've actually shown that in endothelial cells have a preferred I'm going to get to that. A preferred orientation, and it is. Epithelial cells don't show a preferred orientation, but you see the same thing. So, yes, you're right. But it turns out you often get alignment of cells, but even when you don't, you still see the same thing. This looks really complicated, and it is, but it's very understandable. So this is showing the tension. So a tension is a number like pressure, the opposite effect, the negative of pressure. And it's just the average value that doesn't take into account direction. So would you be surprised if I told you, if I looked at the state of stress here, that if the tension might be, or the force might be bigger in one direction than another? You probably wouldn't be surprised because we call that anisotropic stress. So what we did is, at every point, let's say here, here, and here, we can represent the state of stress by an ellipse. So the ellipse represents the stress, and what it's saying is that cell right there is pulling more this way than it's pulling that way. And actually, that major axis of the ellipse is a measure of the stress. So it's very anisotropic. What you can also see is there's a preferred direction. So that one, it's called principal orientation. So those of you who study mechanics, so that's the principal orientation, and you can see that it's cooperative. There are kind of these big swirls of stress that are kind of cooperative over 10 to 50 seconds, depending. And again, here there's these big cooperative regions where the cells are kind of showing like as if they're flowing and trying. You can see that they're in this case they're aligned with the cell orientation, the cell body. Now, what I didn't tell you yet is what the red arrows are. It turns out the red arrows are the migration velocities we measure by particle image velocimetry. So, and what your eye tells you, if you look down here, you can see it very clearly. Your eye tells you that the cells seem to be moving along the direction of principal orientation. they're following the alignment of the ellipses. So I don't know why that is, but it's what we observe. It's very systematic. It's not ironclad, but I'll show you the statistics in a second. Again, here, these cells are swirling this way, and they tend to follow the direction of the stress orientations. So this is the first connection between stresses and motions. It's not a complete equation of motion, but it's an important relationship. Now here's a really interesting thing that we don't understand, but we think is very, very important. For any of you who studied engineering mechanics, you know that these principal orientations are special for another reason, because it turns out those orientations are orientations in which the shear stress is zero. So if you look at, here's a cell, outline the cell. That's a normal stress, normal meaning perpendicular. So this is like this cell tugging on the one next to it. The blue arrow is the shear stress, like this, rubbing against it. So that's the shear stress. And what this picture is saying is cells move along the direction of maximum principal orientation, which happens to be the direction of zero shear stress. We know that, it's for sure. So it's saying that somehow cells, they're happy to pull on one another, but they're not happy to have shear stresses. So somehow the adhesion molecules here that are sticking these cells together, they can't support shear stress. That's a new finding. No one's even thought about shear stresses because you couldn't measure them. And now we can measure them, we can image them. I haven't shown you the image, but the shear stress is actually the ratio of the maximum, the difference between the max and min. And what this says is that cells tend to move along directions of zero shear stress. So this is, it might sound esoteric, but it's really important in terms of how cells move. So along principal orientations, by definition, shear stress is zero. And that says cells track along shear-free trajectories. I think this may be a really important principle of biology. So it's not chemotaxis, it's not aptotaxis, it's not durotaxis. It's something else that's innately collective. It's not about one cell. This doesn't happen for one cell by itself. So we gave it a name. It's good to give things a name. So we called it plithotaxis. That's the Greek. And plith is the Greek word for crowd, swarm, or throng. And taxis means arrangement. So, and the principle is that neighboring cells join forces to transmit normal stresses, the red arrows, across cell-cell junctions, but they migrate along orientations of minimum intracellular shear stress. So this was Don J. Tombay's discovery, and it's getting some attention. So that's good.
Would you explain a simple thing? How do the cells actually apply a force to the layer? Is it legs or
The substrate.
Yeah.
Oh, I didn't say. So there's adhesion molecules, especially integrins. So integrins are transmembrane molecules. They hook, and I also didn't say, these are collagen-coated gels. So the integrins love to bind to collagen in their extracellular domain, and they bind to focal adhesions and stress fibers, cables inside the cell. I probably should have shown you that. So thank you for, I'm going to put that in. So it's those adhesion molecules. Integrins are how cells, even single cells, crawl along. They pull themselves along.
So they like a suction cup, and then
Kind of like it bonds like crazy glue almost. It binds, and then you have.
how do you let it go and to move?
And then it's got motor proteins inside that pull the cell along that cable, and then in the rear of the cell it pulls up those adhesions and keeps moving forward. That's how it does. This is a very highly studied area, and this is pretty well known. And it's also known that in terms of cell contacts, there are three different kinds of adhesions. There are adherens, junctions, which are cadheterins, which are homodimers that hook to each other. There are desmosomes, there are tight junctions. So there's all kinds of molecules here. But apparently they can't support shear stress. They're happy to pull, but if you go this way, they either break or slide or something. They can't exert any force.
But then, for them to go the same orientation and avoid shear stress and something, there must be some control of this. The adhesion molecules.
There must be. And now we have the tools to start to look at it. Because we can measure. Before it was just in the dark. So now the lights are on. So yeah, Yeah, I mean, this is not something I could do, because you have to know about the signaling pathways and so on, which is not my thing. But in terms of the forces, you can do that. So here's kind of like peeling the layers, seeing the phase contrast image and all the stresses. So same thing there. Let me, in the interest of time, I'm going to actually skip over this. These are the statistics that back up everything I told you, and I'm going to skip it, except to say this. We actually tested it on certain lines of cancer cells with certain interventions. I'm not going to go through these details. Let me just skip forward to just the result. Here's the result. These are breast cancer cell lines here, and this is the control. This is an oncogene that causes a proliferation, so you can see there's more cells here. And importantly, this is another oncogene, 1433 zeta, that breaks down cell-cell junctions. And what you find is that in this case, this is showing how cells move relative to the principal orientation I told you about. So cells get steered by their neighbors, and they're just like at the hockey game. You wind up getting channeled by your neighbors, even if you don't want to go that way. So there's some kind of a control here or steering, but when you disrupt cell-cell junctions, you can go in any direction. The principal orientation is this way, but you go that way. So this is, we think that here, plithotaxis is strong, so there's strong cell-cell guidance, but here, when you get a metastatic kind of disease, you lose cell-cell guidance, and cells can just go scattering off all over that time. So let me skip that. Okay. I'm just going to do this next part, and then we'll stop. So what I've told you so far is that there's this plithotaxis idea, and it's kind of intuitive. It says that cells migrate in the direction that they are pulling themselves. Okay. Well, that makes sense. And also in the same direction that they're being pulled by their neighbors. So on that's what we find, that's what plithotaxis is. And it's the tug of war idea plus the idea that you get steered by there's no direction with you get highly steered by your neighbors. And there's this corollary that I mentioned that it means cells migrate in direction of minimum intracellular shear stress, which is something I think will keep a lot of people busy for a while. But now there's and then those are those stresses. Now I'm going to jump into it on something this is now brand new. It's not published yet. And actually, this is the first time these data are being presented. So hopefully this won't be rough. The idea across. So you're the guinea pigs. It's something that we call the Aquino Axis, and this is totally counterintuitive. We were shocked, and we weren't even trying to find. We were trying to do something else, and we stumbled into it. That's the way it is. So when things don't work out the way you thought, that's when you really discover something interesting. This is one. And it says the cells systematically migrate 90 degrees or even 180 degrees from the direction they're pulling. So I'm going to pull this way. I'm going to go back to this. Let me show you how this works. So this has, I call it edge states of Aquino Axis. So here's our friend, the MDCK cells. Here we did something very clever. We were impressed by everything being heterogeneous and fluctuating. And I had said to the fellow, Jay Kim, a bright Korean guy with background in engineering, didn't know any biology. I said, we want to get the equation of motion. Like for those of you who are mechanics, Navier's equation, Navier-Stokes equation, what's the relationship between force and velocity? And it's going to be really hard with all these fluctuations going on. So instead, why don't we put an obstacle? So this is an obstacle. Let's put an obstacle in the way of the cells so that we get systematic gradients. And then we're going to even make bunches of these, and we'll average over them. So we average out the fluctuations. So we'll get the smooth motion of the cells, we'll get the stresses, and we'll discover the relationship between the motions and the stresses, the equation of motion. That was what we wanted to do. It isn't what we did. So these are MDCK cells. I'm going to play the movie in a second. They're coated on, this is collagen coated gel, but here we made a mask where there's no collagen here, so the cells can't stick. The the cells can't go. They might want to go here, but they can't go here. So we call this a frustrated edge, because the cells kind of want to go there, but we don't let it go there. So let me play the movie. Play a movie, and we'll see what happens. Okay, there go the cells. There they go. Oh. Sorry, you have to watch it twice. Okay.
They don't reflect back.
No, they don't. Although they send, it's a whole different story. So there's stress waves that bounce back. I'm not going to tell you about those. We published that. I'll tell you what. So in your mind's eye, in fact, what we were thinking is, you can imagine the cells, you see them moving, you imagine that they're pulling themselves along in the directions of motion. So that's a plausible idea. It fits our intuition. It's even consistent with Plithotaxis, and it turns out it's totally wrong. So let me show you why I think it's wrong. So now back up. Here's the monolayer of MDCK cells. Here's our region that they can't stick to, bare gel. And this is a little while later. They've enveloped part of this barrier. Now they've totally surrounded it after 24 hours. So let's look a little closer. These are the velocity vectors, the migration velocity. So here you can see there's lots of fluctuations. They hit the barrier. That's what engineers call a stagnation point. That's where actually the cell velocity is 0. There's no motion normal to the surface or parallel. So the stream divides this way and this way. Here's this region over the top. And now here, that's a downstream stagnation point. The cells have come around, stop, and then turn. So you get an upstream and a downstream stagnation point. So this is for one realization. And then if you average over a bunch of these things, we averaged over 6. It turns out we should have done more, but we did 6. It smooths out the fluctuations. So you see all the cells heading on average from left to right. Here they are. Here's the stagnation point at the equator. Here's the downstream stagnation point. So you get nice velocity profile. So any of you who studied fluid mechanics, we flow over a cylinder. Same kind of problem. Okay. Here are the traction forces. These are the forces exerted by the cells on the gel under them. So blue represents cells that are pulling themselves to the right. And red are ones pulling, you see a few here, pulling the other way. But again, this is average. So on average, it's very much monolayers pulling itself to the right. It collides with the barrier here. I'll come back to this one. But here, look at this. So this is now in the downstream part, where after the cells converge, and it's not blue anymore, it's red. So these cells are actually moving this way and that way, I'll show you, but they're actually pulling to the left. And actually these over the top, this is the traction in this direction. This one is, they're pulling down. So this is really strange. So the cells always pull towards the empty space. Let me go into a little more detail. This is actually, the color code here represents tension in the monolayer. So the tension starts at 0 and it builds up going away. And now we can, this is a blow up of this region. So far away, before you hit the barrier, the stress orientations at, which are the ellipse, the velocities, which are the blue arrow, and the traction forces, which are the black arrow, are all aligned. That's like Plithotaxis. But here, something else is happening. Okay, let me blow these up. Look what happens here. When you get to these, the cells are pulling this way. The principal orientation is that way, and the velocity is this way. They're systematically misaligned. This is very strange. And look over here. If you look at over the top, take, let's say, this one. The cell is pulling this way. The principal stress orientation is that way, but it's moving that way. And here the cells, right near the surface, they're pulling towards the empty space. And here, even though the cells are moving away, they're pulling back. So this seems crazy, but it's actually not so crazy. Anyone here sail? Any sailors? Okay, so look at this picture. Which way is big?
In Winnipeg?
Well, there's a lake. There's a lake. Which way is the wind coming from?
West.
In this picture. In this relativity. So you're the sailor. Can you tell which way the wind's coming from? Right.
Yeah.
No, it actually is coming right. It's coming right. Like forward. This is close haul, and this is beating to windward. So it's counterintuitive, but it works. You can take a sailboat, and the wind is going this way. You can sail into the wind. It's called beating to windward. You can come to about 45 degrees. So how can it be that the wind is blowing this way, and you can sail into the wind? No, you can't. It turns out if you think enough about it, you can watch it happen, and you can actually figure out the physics. So this is the same kind of thing. We don't understand it, but somehow cells, let's say cells here, they're pulling to the left, but they're moving to the right. 180 degrees different. Well, let me skip that. And so again, here, these cells are all oriented to pull towards this empty space. So we call this kenotaxis. We don't understand it. Keno is the Greek word for vacuum. So this is vacuum arrangement. It says that local tractions polarize towards unfilled space, or what we call the frustrated edge, regardless of how they're moving. So the cells just, when they see unfilled space, they want to go there, but they can't, because they can't stick, they can't walk on that. So somehow kenotaxis trumps lithotaxis. The cells, somehow they can pull one way and set their intracellular stresses another way and go third way, kind of like a boat sailing upwind. And we don't understand it, but the data are very, very solid.
But it tells you that the control system, whatever motion mechanism there, is quite stupid. Because, I mean, here is a barrier that you can't cross, but you just keep.
Yes, although it's saying that these monolayer cells, they're programmed to fill empty space, like the heal a wound, fill a gap.
You think they should stop.
You think they should stop, but if they don't. So this is how they behave. So we need someone smart here to figure out how this works. Actually, I'm going to let me go on. Let's see. So there's one more thing about this, and then I'm going to stop. I think I'm going to just about out of time. This would be a good place to stop. So I noticed here, I said, this is crazy, because okay, the cells are pulling to the left, but they're moving to the right. But then if you go way downstream, everything realigns. Now they're pulling and moving in the same direction. But this system is totally symmetric, so somehow the traction forces have to flip, and they could flip counterclockwise or clockwise or counterclockwise. They could go either way, but there's no way to tell. If you go like a coin balanced on its edge, it could go one way or the other. That's a sign of instability. Right? The coin on its edge is unstable. So this is going to be an unstable flow, like a turbulent flow in fluid mechanics. So I said to our guys, there's got to be something weird happening here. So we started to look closer. And lo and behold, we did a bunch of different imaging. So this is showing tight junction proteins. This is just showing cell shape here. This is eccentricity of the cell. This is cell area. This is showing cell boundaries. This is the orientation of the cell. And this is major axis length. And sure enough, right at that point where you get the downstream stagnation point, you can see that just something weird is going. It's just the cells are they're somehow flipping their tractions around, and cells that are, let's say, right there, they're pulling to the left, but their neighbors are pulling them to the right, so they get stretched like taffy candy. Right? The intracellular, their neighbors are pulling them one way, but their feet are going the other way. So you get these weird shapes, and this is showing, let's see, this is actin, and this is CO1. And it was interesting. I showed this to some of the people in my program who are pathologists, and they said, "Oh yeah, this look this. I think I put it in here. These look just like what's called foreign body epithelial cell granulomas. I don't know what that is, but he said that you see these whenever you have near sutures or microimplants. So it seems like this isn't just an artifact of the culture that capture something that's actually going on in the body. So actually, you know what I'm going to do? I'm going to stop there. Good place to stop. And I'll take any questions that you have.
So what's the next step?
The next step. We'd like to understand some of the mechanism of what's going on here. So actually, this one which I didn't get to. There's something called Cryptic Lamellipodia. So one of the ideas about how cells move in a cluster is that they extend a foot-like structure under the cell in front of them, and the thought is that that's the direction they move in. And here, what we've shown is that actually it doesn't work that way. That the cells could have the lamellipodia this way, but they go that way. So but that's not really the next step. The next step is we're trying to understand, actually in particular, how these kinds of assays can be used in particular diseases. So I don't know if you know Lisa Hensky. So she called me about a year ago. She studies two lung diseases, Birt-Hogg-Dubé syndrome and another disease called LAM. Can you tell me can you say LAM is this big one?
Lymphangiomyomatosis.
That's it. Okay. So these devastating, LAM is a disease that afflicts young women, and it's devastating. And she had reason to believe that there's a particular adhesion molecule of cell-cell contact that's deficient or defective in these people. And she wanted to know with these kinds of assays, can we learn something about what's going on? So now we've got a big LAM and Birt-Hogg-Dubé syndrome, where we're going to use these assays to try to understand how, in the case of LAM, kidney cells migrate to the lung and it's a cancer-like disease, but it's not cancer. We're trying to understand the nature of the disease and how the cells move. So that's one. We're probably going to take on, actually, the problem is there are too many things for us to do now biologically. There's issues of cancer, cell scattering. How does a cell pull away from the pack and metastasize? Well, that's another thing we're interested in. There's a bunch. And now we kind of now that we have these assays and we've just got this thing I just taught you in review, we're going to start turning back to the biology and try to use these to answer some
Must be quite relevant to invasiveness of tumors.
It's got to be. It's got to be. And I'll tell you something. This is really interesting because Lisa Hensky was shocked by this. The traditional method that she was using to measure cell-cell adhesion was you take a bunch of cells, and whatever, these were kidney cells, but they're also known as lung cells, and you suspend them in a droplet on the bottom of a dish, and they cluster together. And then you shake it, and the cluster will break apart if you shake hard enough. That's the adhesion axis. Pretty crude. So we took the same cells and we put it in our system, and she came to the conclusion that when folliculin is the molecule that she had discovered it has a defect, she had found when folliculin was defective that those cells were more adhesive, which may sound strange because it's an adhesion molecule. But anyway, and she wrote a paper saying that they get more adhesive. Well, why would you have trouble with cell scattering if they're more adhesive? Well, we put them in our system, and we found that those cells can't transmit forces from one cell to the other in these assays. So the adhesion's less. It's just kind of an artifact of that assay. That there's no matrix for the cells to bind to, and it's the wrong assay. But this is the right assay. So I think there's going to be lots of things like this. And now that we have the lights on and we can see all these forces, I think there's going to be. I need more people and money.
Well, you need people who work with both
Right.
to figure out how they control.
Exactly. Those are the next things. The other thing we're doing is issues of transition to three. These are all two-dimensional assays. What happens in 3 dimensions with cell clusters in 3D? We're working on
You should have met John Burman here. He's a great adhesion.
Oh, really? Okay. Well, next time.
I have a question. Do you think that there may be macro level forces that are creating downward causation?
I didn't understand.
Maybe there are
Macro level
properties or forces that are creating downward causation on the cells? like system properties that are.
Oh, yeah, for sure, because you get these.
Coming back.
Yeah, well, you get, first of all, it turns out, I didn't show it, but we published this, that if you have a cell island, the tension grows from 0 at the edges to big values inside it, and cells are mechanosensitive. So this collective, this tug-of-war phenomenon changes the whole tension level in the monolayer, and that has to be signaling, turning genes on, turning genes off, and that's a collective, integrated level thing. And another thing that we're also doing, one of the fellows is, in terms of the endothelial cells, we hadn't actually put fluid flow or simulate blood flow, and in terms of that's having big effects too, although we haven't organized our thinking about that. So I think there's probably a lot of collective things like that are going on, but we really haven't looked at them yet.
Oh, sorry.
No, go ahead.
Yeah.
Okay. I'm just a bit concerned about the result that referred to because when we try cell migration, let's say in airway smooth muscle cells in a pathological context like asthma, like individual cell migration shows huge variety in the result. Then I don't know this collective cell migration approach, how it will be addressed in vitro using this approach.
How it will be approached
Because in cell migration we usually have a lot of variations in results, especially in pathological context.
Right.
I have no idea about endothelial cells, but for ASM cells, I think that
In terms of the variation.
Exactly.
Right.
I don't know how is the reproducible having the same kind of results following this kind of chemo axis.
Yeah. Well, I don't really know the answer, but we have a hypothesis about it. We've alluded to in some of our papers, and I have a review paper that was just accepted. And it has to do with this. We think that part of the variation has to do with this jamming phenomenon, the fluctuations that I emphasized in the beginning. And if it is jamming, it turns out it unifies a whole bunch of things, because you can imagine a phase, I don't want to get too thermodynamic for you, but you can imagine a phase diagram where you can imagine cells being glommed together and jammed if they're held together by superglue. So if adhesion goes up, they can't move. If they're too crowded, they're crowded together like mad, they can't move. If their motile forces are attenuated, they can't move, and the reciprocal of all of those. So we have reason to think that actually everything gets unified in a jamming phase diagram. So kind of like freezing, and it would carry over even from 2 dimensions into 3-dimensional systems. So this whole idea of jamming would span the dimensionality of the system, and we'd be able to account for everything in the context of a phase transition to jamming. All the features that we saw, including smooth muscles, at least in culture, tend not to. They can get crowded, but they don't make cell-cell junctions.
Exactly.
And so you get another form of volume exclusion, right? If a cell is migrating, it can't go through another cell, just like coffee beans in a chute. So, but that causes jamming. So I think a lot of the variation you're seeing could be because you're getting close to what we call a jamming transition. I can give you the paper that you have on that, but I think that may be something that unifies everything.
And the interesting thing is that it happens mostly in asthmatic cells, like in this case, compared to healthy normal ones.
Well, people are really interested in migration of smooth muscle cells. That's kind of like a whole bunch of people are interested in that. Anybody else? It's okay, guys. There's no such thing as a dumb question.
Is it feasible to have a layer of the fluorescent spheres, then the culture, and then another layer of some fluorescent spheres?
We've never done that. Some people have started to do that, to make sandwiches. Actually, there's a guy, at Brown University, that's right, Christian Frank, is doing just that, and he's made like a double traction microscopy with 2 layers, and he puts a cell in between, and then he squishes it so the cell sticks to both sides. And so, can it be done? Yeah, it's being done. I don't know that he's published it yet, but I was in his lab and he was doing just that. So that gets you closer to, it's a little bit away from the 1D. It's kind of more 3D. But yeah, for sure. Okay, well, thanks for listening.